When we talk about saving coral reefs, the picture that often comes to mind is maybe helping out those faster growing branching corals.
That's right. They get a lot of the attention.
And they are important, absolutely. But today we're going, well, deeper. We're looking at a really crucial, but maybe sometimes overlooked part of reef recovery, the massive corals.
Yeah,
Yeah.
the big ones, the foundation.
Yeah, the ones that really build those incredible underwater cities we picture.
Exactly. These are, fundamental to the whole ecosystem, the framework. These large stony corals create these really complex 3D habitats.
Homes for so many creatures.
Oh, absolutely. It's estimated like 25% of all marine fish depend on these structures. Think about that.
Okay.
A quarter of all fish species in the ocean rely on coral reefs, and these massive corals are a huge part of that structure.
That's just staggering. And it's not just about providing shelter, is it? There's more to it.
No, not at all. They're vital for, well, for making sand for our beaches, for one thing, and they act like natural breakwaters.
Oh, right. Damping wave energy.
Precisely. They protect coastlines from storm surge and erosion. I mean, they do so much for the ocean, and frankly, for us too.
So, given how important they are, why the focus on the faster growing ones in restoration? You mentioned that they're often overlooked.
Yeah, it largely comes down to that growth rate.
Yeah.
Massive corals are naturally very slow growers. So restoration efforts often prioritize the species that showed quicker results. It felt more achievable, I suppose.
Like trying to regrow a redwood forest versus planting fast-growing bushes.
Kind of, yeah. It felt like a daunting, incredibly slow process with the massive ones.
Okay, so let's talk about those traditional methods for a second. What were we typically doing, and why wasn't it really cutting it for these giants?
Well, often it involved taking fragments, small pieces of coral, maybe from a healthy reef, and transplanting them onto a damaged area.
Okay.
And look, these efforts weren't without success, especially locally. But for large-scale, ecologically sound restoration, they had limitations. A big one was just sourcing the material.
Where do you get the coral from?
Exactly.
Hmm.
If you take too much from existing healthy colonies, you can actually harm them, reduce their own ability to grow and reproduce.
Right. You don't want the solution to become part of the problem.
Precisely. And then there's the issue of what happens to those small fragments once you put them out there.
Vulnerable?
Very, especially the smaller ones. They can be quite susceptible to predation. Think parrotfish, for example.
Mm.
They sometimes seem to find these little nubbins an easy snack.
Ah, so you put out these tiny, hopeful fragments, and they just get eaten. Not exactly efficient.
Not ideal, no.
Yeah.
But this is where things get really interesting because scientists have been, thinking outside the box, finding ways around these problems, especially that slow growth issue.
And that brings us to microfragmentation. I've heard this term. It sounds like a potential game changer. What is it?
It really is fascinating, microfragmentation. Well, the name gives it away a bit. You take a larger, healthy coral colony, and you break it down into lots and lots of very small pieces,
Microfragments. How small are we talking?
often just a few square centimeters. Tiny.
Okay, tiny pieces. My first thought is, wouldn't that make them grow even slower? Or be more vulnerable?
That's the incredible part. It's completely counterintuitive. These microfragments actually show dramatically accelerated growth rates.
Faster. How much faster?
In some studies, up to 40 times faster than they'd grow naturally out on the reef.
Whoa, wait. 40 times?
40 times, yes. It's like it triggers some kind of rapid healing and growth response. The coral puts all its energy into expanding its tissue area quickly.
That's just incredible. What's going on biologically? How does that work?
Well, remember that corals are clonal. All the fragments from one parent colony are genetically identical.
Right, like clones.
So when you place these microfragments from the same parent close together, say on a tile or a plug in a nursery,
Yeah.
they grow outwards, their tissues meet, and because they recognize each other as self, they fuse together seamlessly.
Ah, so they join forces.
Exactly. Instead of competing, they merge. So you end up creating a much larger single piece of coral, much, much faster than if one fragment just grew on its own.
That makes so much sense. It's like a shortcut to getting a bigger, more robust colony started. Have we seen this work well with specific massive coral species, the really important ones?
Oh, definitely. There's some great research published in PeerJ, for instance. They looked at Orbicella faviolata, that's Orbicella faviolata,
Okay, Orbicella faviolata, got it.
a really key reef builder in the Atlantic. Using microfragmentation and fusion, they saw growth rates up to, get this, 63 square centimeters per month.
63 square centimeters in a month. For you listening, the learner, That's like watching a dinner plate-sized area of coral tissue grow in less than half a year from tiny fragments. That's wild compared to natural rates.
It is. And another important Atlantic one, Pseudodiploria clubosa.
What?
Pseudodiploria clubosa.
Yeah.
Showed rates up to 48 square centimeters a month,
Hmm.
even in the Pacific, with species like Porates lobata, Porates lobetu.
Porates lobata. Porates lobata.
They've seen rates up to 23 square centimeters per month in fusion experiments, sometimes even higher depending on the exact conditions.
These numbers are just transformative, aren't they? When you think about the usual growth being millimeters per year.
Absolutely game-changing for restoration timelines. And interestingly, the research also seems to suggest that starting with slightly larger microfragments can lead to even faster growth.
Okay, good to know.
And crucially, these corals grown through microfragmentation and fusion have been observed to spawn later on.
Ah, so they can still reproduce normally. That's vital.
Exactly. It suggests this speed-up process doesn't mess up their fundamental life cycle, which is obviously critical for long-term reef recovery. We want functioning reproducing reefs.
Definitely. Okay, so microfragmentation itself is huge, but I imagine there are other factors in these nurseries that scientists are tweaking to get the best results.
Oh, absolutely. It's not just about chopping them up small. The environment they grow in matters immensely.
Like what specifically?
Well, fragment size itself, even within microfragmentation, seems to play a role. Research in the journal Aquaculture looked at this with Porates lobata and another species, Porates compressa, Porates compressa, Porates compressa. Okay, two Porates species. What did they find about size?
It was interesting. For P. lobata, the smaller fragments actually started off growing a bit slower than slightly larger ones. But for P. compressa, the growth rates were pretty similar across different small sizes, unless predators got to them.
Huh. So it can be species-specific even there.
It seems so. Different species might just respond differently, maybe how they allocate energy reserves when they're fragmented. We still have lots to learn there.
But the good news...
The good news was that even the really small nubbins had high survival rates in these controlled land-based nurseries, like 70 to 90%, and they still grew significantly. So starting small is definitely viable, especially if you can protect them early on.
Right, protection is key. And the nurseries themselves, are there different ways to set them up? Yes. Researchers are comparing different approaches. Some are quite high-tech, high-maintenance, with really detailed monitoring.
Okay.
Others are testing simpler, lower-maintenance systems. Sometimes they even introduce things like sea urchins, Trypnustes gratia.
Sea urchins, Trypnustes gratila, to do what? Eat algae.
Exactly, like little janitors to help keep algae from overgrowing the coral fragments. It's a clever idea, using natural grazers.
But I sense a but coming.
Well, yeah, there's a trade-off. The low maintenance is good, saves effort and cost, but you run the risk that the urchins might not just eat the algae, they might also accidentally graze or dislodge the tiny coral fragments, especially the really small ones.
Ah, okay, a delicate balance then.
It really is, finding that sweet spot.
What about things like light and water flow? Basic environmental stuff. Do different massive corals have different tastes, so to speak?
They definitely do. This was explored in the Journal of the Marine Biological Association. They looked at Montipora capitata, Montipora capitata,
Montipora capitata.
yeah, Porates compressa again, and also a branching type, Bacillopora demicornis. Bacillopora demicornis, just for comparison.
Okay, what were their preferences?
It varied. P. compressa, the massive one, seemed to do best in highlight, but with low water flow.
Interesting. Highlight, low flow.
Yeah. But M. capitata, another stony coral but maybe less massive, actually preferred more shaded conditions. And sort of interestingly, across the board, really high, constant water flow seemed to result in slightly less growth overall compared to low, constant flow.
So blasting them with water isn't necessarily better.
Apparently not for these species under these conditions. It really highlights that you need to tailor the nursery environment to the specific needs of the corals you're trying to grow. It's not one size fits all.
Makes sense. Now, that same study also looked at feeding them artificial food. How does that even work for coral?
Yeah, it's another fascinating angle. Corals aren't just plants with algae, they are animals, and many can capture food particles from the water. So researchers tested various artificial food supplements.
Like little powdered fish flakes for corals?
Sort of, more scientifically formulated, but yeah, the idea is providing extra nutrition. And they found, again, it varied.
How so?
Some of the foods boosted growth in M. capitata and the branching P. demicornis, But those same foods didn't seem to help P. compressa much at all.
So P. compressa is maybe less reliant on capturing external food, more on its algae.
That could be it, or maybe it just didn't like the food offered. It suggests different feeding strategies among species. And another key point, more food wasn't always better.
Oh.
In some cases, increasing the dose of the artificial food actually led to decreased growth.
Too much of a good thing, maybe. Or maybe it fouled the water.
Could be either or both.
Yeah.
It suggests there's an optimal level, and just dumping in more food isn't the answer. You need the right balance.
Finding that perfect recipe. One of the things that really surprised me in the research was about, well, managing the gunk that grows in the tanks, the algae on the sides and stuff. How does that affect coral growth?
Isn't that interesting? Yeah, another Piaget study dug into this, specifically looking at Porites compressa microfragments again. They focused on how different algal communities, the stuff growing on the tank surfaces, influence coral growth in land-based nurseries.
Okay, what were the different communities they tested?
They had three main approaches. One called mini reef, trying to mimic a diverse natural reef surface. Another called clean start, where they really encouraged beneficial crustose coralline algae, that hard pink stuff you see on healthy reefs.
Right, CCA. Got it.
And a third, and this is the really surprising one, was called green film. They basically let or even encouraged a thin film of, well, simple green algae to grow in the tanks.
Green film, like the stuff people usually try to scrub off their aquarium glass.
Exactly. Sounds like the opposite of what you'd want for optimal coral growth, right? You'd think it would compete with the corals.
Totally. So what happened?
Well, here's the kicker. The green film method, especially during the cooler winter months in their study, actually resulted in the fastest growth of the Porites compressa microfragments.
Get out. The green slime tanks grew coral fastest.
That's what they found. And the corals in those tanks also had the highest health scores overall.
That is completely counterintuitive. Why? Any ideas?
The hypothesis is that this particular type of benign green algal film might actually help the corals by outcompeting other, more harmful organisms.
Like what?
Like, say, cyanobacteria or more aggressive turf-forming algae that can really smother or irritate coral tissue. So the green film acts almost like a protective layer, preventing the worst stuff from getting established.
So it's not about having a perfectly sterile environment, but maybe cultivating the right kind of friendly neighbors.
It seems that way, at least in this context, for
Yeah.
P. compressa. It's a really insightful finding, showing just how complex these interactions are, even in an artificial tank environment.
Wow. Okay, so we have microfragmentations speeding things up dramatically, and
Yeah.
all these factors, fragment size, nursery type, light, flow, food, even the algae on the walls being fine-tuned. Let's zoom out. What are the real-world applications? Where is this heading?
That's the crucial question, isn't it?
Yeah.
How do we scale this up and make a difference on actual reefs?
Right.
The potential for microfragmentation and fusion for large-scale restoration
Yeah.
is, well, it's enormous. We're talking about the possibility of rapidly generating significant coral cover on reefs that have been badly damaged.
Re-skinning the reef, almost?
In a way, yes. And people are thinking creatively. There's talk of using these techniques to build living breakwaters.
Living breakwater.
Yeah, imagine attaching these fast-growing, massive corals, propagated using microfragmentation, onto artificial reef structures or existing seawalls. You could essentially create coastal defenses that are alive, that can grow, self-repair to some extent, and provide habitat.
That's blending engineering and ecology in a really powerful way.
It is. And beyond just growing corals for planting, these rapid growth techniques are also super useful for research. They allow us to develop standardized growth assays, meaning we can have reliable, quick ways to measure how corals respond to different things. Potential stressors, like warmer water or pollution, or maybe beneficial treatments. You get answers much faster than waiting for years for natural growth.
So better tools for understanding coral health and threats, too.
Exactly. And of course, there's ongoing practical work. How do you best get these nursery-grown corals to stick onto the reef substrate long-term? Improving self-attachment methods is a big focus.
Makes sense. They need to stay put once they're out there.
Definitely. And we're seeing more integration, combining the strengths of these controlled land-based ex situ nurseries with efforts happening directly on the reef, in situ, for really large-scale propagation.
It sounds like a much more proactive, multi-pronged approach than just trying to protect what's left.
It is. And looking even further ahead, people are exploring concepts like assisted gene flow. That involves potentially introducing genetic diversity. maybe from corals that have shown resilience to heat waves in one area into populations in another area to try and boost their overall ability to cope with future climate change. It's complex, ethically and logistically, but it's part of the conversation.
Wow, so it feels like we're really shifting gears from purely conservation to active restoration and maybe even enhancement.
I think that's a fair way to put it. The key takeaways here, especially for you, the learner, are really twofold. One, microfragmentation is a genuinely powerful tool that helps us overcome that huge barrier of slow growth in these critical foundational massive corals.
It really changes the timeline.
It does. And two, it's not just one magic bullet. Optimizing growth involves this whole suite of factors: the fragment size, the nursery conditions, light, flow, food, even, amazingly, the type of algae in the tank. They all play crucial roles.
And hopefully, this deep dive gives you that aha moment, seeing how scientific discovery and, frankly, a lot of careful experimentation can lead to tangible ways to tackle these really big environmental challenges. The idea that we can speed up nature, in this case, the growth of these vital corals, it really opens up exciting new avenues for genuinely effective reef restoration.
It certainly does.
Yeah.
Which I suppose leads us to a final, maybe provocative thought to leave you with. Given these rapid advancements, this acceleration in our ability to cultivate and grow corals, could we be perhaps on the cusp of a new era? An era where we can not just try to patch up reefs, but actively rebuild and maybe even enhance coral ecosystems on a really significant scale? And if so, what does that mean, and what are the responsibilities that come with that kind of power?
A lot to think about there, the potential and the caution needed.